Good morning and welcome. Thank you for joining our webinar today on an introduction to PBBK, Simcyp Discovery, and the Simcyp product suite. My name is Mark Lovern. I'm the Senior Vice President of Product and Commercial Strategy for Certara's Simcyp Business Unit, and I am joined today by my colleagues Himanshu Mishra and Kevin Romer, both will respectively Director and Vice President of Business Development. Before we get started with our main presentation, I have just a couple of housekeeping items. Our main presentation should be about 45 minutes long, which should leave ample time for question and answer. And we will essentially we will provide input. You will see a demo, and towards the end of the webinar, you'll have the opportunity to ask questions. Actually, you have the option of submitting questions at any time through the chat window. If you experience any technical issues whatsoever, please message us through the chat. And as a reminder, today's webinar is being recorded. We will email you a link with the recording in a few days. As I mentioned before, you're welcome to submit questions at any time during the presentation, but all participants will be in listen-only mode during this webinar. So without further ado, it's my pleasure to start our presentation. Well, actually, as I said, introducing Kevin and Himanshu, and we'll be back over to you guys shortly. First, I'm going to just do a few introductory slides about PBBK and the Simcyp Discovery and our product suite. So for those unfamiliar with physiologically based PK, otherwise known as PBPK, it has a relationship with classical PK. As many are familiar, classical PK models have been around since probably the late 19th century, early 20th century, and they are in common use throughout pharmacology. One of the primary purposes of classical PK models is to describe the concentration time course of a drug in the central blood pool. As such, most of the main features of the model classical models do not have what I would call a direct physiologic interpretation. So the peripheral compartment is largely an empirical construct that's introduced into the model to essentially mimic certain behaviors of the drug in the blood pool, most notably that it exhibits a two-compartment behavior. But the peripheral compartment itself does not have an interpretation in a physiological sense. Well, as the name suggests, for physiologically based PK models, they essentially take a different approach whereby each compartment does have a physiologic interpretation. So as you can see on the right, you have compartments for lung, adipose tissue, bone, brain, etc. Another feature of physiologically based models is that all of the transport of the drug is mediated through blood flow, which is not necessarily the case for classical models. So, essentially you cannot get a liver metabolizing a drug before the blood has transported the drug to the liver. Now, one might ask oneself, what is it that these models bias? They have considerably more complexity. But the answer to that question is the greater complexity also brings with it greater predictive power, so that you're able to make more robust predictions to circumstances for which you do not yet have experimental data. As such, these models are often used for first time in human dose selection, predicting drug-drug interactions, and extrapolating to other populations, including pediatric as well as organ impairment. So essentially, the PBBK approach may have benefits throughout the drug research and development cycle, starting very early on and moving on into later development. And what changes over time are the nature of the questions of interest and also the data you have available. So very early on in preclinical development, you may have only in vitro data or maybe some animal data, and your question might be, well, what's the best dose to introduce into humans? So the data is sparser, and the question is one mainly of translation. As you move through the development process, those questions may shift to things such as, well, is there a food effect? Or is my, what's my best formulation for using in humans? Or is there some possibility of drug-drug interaction? And, to support the answer to those questions, generally you're going to have more data. You might have clinical data in humans at that stage. And as the program progresses, you will accumulate more data that can be used to validate or qualify the model. But in addition, as you learn about the drug, there will be other questions that arise, including as we get later into the program, potentially extrapolating to special populations. In general, there are three areas of impact which generally the PBBK applications fall under. So one is to actually apply the PBBK modeling approach to obtain waivers to not have to do clinical trials. And many of these are affiliated with drug-drug interaction studies. But other major applications include extrapolation to special populations, including pregnant mothers, organ impaired, geriatric, and pediatric. In some instances, it is not possible to make robust predictions to the extent that one can completely obviate the need for a clinical trial. However, even in such instances, it's entirely possible to greatly reduce the sample size by using a learn and confirm approach based around a PBBK model. So for instance, in an organ impaired population, I might be able to reduce the number of subjects in each impairment category from 6 to perhaps 3. So there is benefit to applying PBBK models, even in the instance that you can't completely avoid the trial. Having said this, there is a growing body of experience in which our technology has been applied to obtain label claims using Simcyp technology in lieu of human clinical trials. And we have over 100 novel drugs that have benefited from that approach, totaling over 300 label claims. Once you've applied the PBBK approach and developed the model, you can use that model to answer multiple questions and make potentially multiple label claim attempts. And so in total, there are over 300 claims based on our technology benefiting those 100 drugs. So to give a little bit of an overview of our product suite, currently we have 3 currently marketed Simcyp applications. We have the Simcyp Animal, the Simcyp Human Simulator, and then Simcyp Discovery is our latest release, which came out last year around this time, and combines some features from both Simcyp Animal and Simcyp Human Simulator. So most notably, you can simulate mouse, rat, dog, monkey, and human all in the same Simcyp Discovery platform. It is a small molecule simulator, and for humans, we offer a healthy volunteer population. But if for large molecule, that would fall under the domain of the Simcyp human simulator. You have the ability to estimate parameters, and we have included a static DDI calculator to allow users to make an early assessment of potential drug-drug interaction risk. And we'll talk a little bit about that in more detail later. We include a advanced dissolution and absorption model, a very highly mechanistic model of the GI tract, which I'll talk about here shortly. And in addition to the minimal and full PBBK model, Simcyp Discovery offers classical 1, 2, and 3 compartment distribution models. We do include a capability for processing large batches of compounds. As we'll talk about, one of the intended applications is for pipeline optimization and formulation selection. And so that batch processing is designed to help with processing multiple compounds at the same time. Further, we support in Discovery the IV and oral routes of administration, which is also supported in Simcyp Animal. And in the full Simcyp human simulator, we offer several other routes of administration, including inhaled, dermal, subcutaneous, and synovial joint. So just a few words about some of the features in Simcyp Discovery, starting with the atom absorption model. Most people are familiar with the first order absorption model, which is the absorption formula that's used in most classical oral absorption models. And essentially, it's essentially a simple exponential flow, fractional flow from the GI tract into the central circulation. Well, generally speaking, the GI tract is not a well-stirred tank, which is sort of what's assumed in the first order absorption model. Instead, it is a segmented organ, each of which segment has its own unique characteristics with regard to pH, luminal volume, permeability, etc. And that's exactly what we're doing in the atom model, is we're segmenting the GI tract and simulating the passage of the drug through that GI tract and its relative affinities and absorption through each of these segments of the GI tract. As I mentioned, we offer both the full distribution model, the full PBBK model in Simcyp Discovery, as well as a minimal PBBK model. The minimal PBBK model really focuses again on only key organs with regard to the drug. So again, the GI tract where it is absorbed, and for that you may use the atom segmented model. From there, via the portal vein, the drug goes to the liver and then generally is observed in the systemic circulation. There's also a adjustment compartment, somewhat analogous to the peripheral compartment in a classical PK model. But again, as with the full PBBK, transport throughout the body and distribution is governed by the blood flow. So that is still there are still physiologic aspects of the minimal PBBK model that are not included in classical PBBK models. As I mentioned previously, we do include a static DDI calculator in our Simcyp Discovery package. You can look at this as a screening tool. It is very conservative, and it's based on regulatory guidelines published by the FDA, EMA, PMDA, and now ICH. It does support both basic and mechanistic static DDI predictions, but you should really view it as a screening tool in the sense that if it says that there's no risk of a drug-drug interaction, that's generally a pretty safe ground that that's not going to be an issue. If it flags a potential interaction, then you're probably going to want to go dig a little deeper and understand the potential magnitude and even if that is that potential is real. So one of the major goals of the Simcyp Discovery Simulator is to facilitate and expedite the transition from preclinical to clinical. And one of the ways that we hope to help with this is by providing a package, a software package that is forwardly compatible. By that, I mean that models developed, human models developed in Simcyp Discovery may be imported into our full simulator. So that models, the PBBK models developed very early on in drug development can then be reused and support the decisions later in development. For instance, again, to assess drug-drug interaction potential, or extrapolating to special populations such as pediatrics or geriatrics. But the key is that you do not have to rebuild a model developed in another platform to be used in the Simcyp full human simulator. So we really developed Simcyp Discovery with 4 major use cases in mind. Most notably, first in human dose prediction, as I mentioned before, compound triage and pipeline optimization, early screening of drug-drug interaction potential using the static DDI calculator, and also early formulation simulation and the exploration of in vitro characteristics that would have an impact on the formulation performance. And at this point, I am going to turn things over to my colleague Himanshu, who is going to give us a demonstration of Simcyp Discovery.
Thank you, Mark. Let me know if you can see the Discovery screen.
Yes, I can. Yes, we can. Thanks, Himanshu.
Okay, thank you. So as Mark said, I'll quickly run a quick demo of the Simcyp simulator. So this is how the screens look like when you open up the application. There are 3 key elements of when you are running a simulation within Simcyp. You have your systems data, or the species data as we call it. So here, as Mark said, we have 5 different species. So 4 animal, you have your dog, monkey, mouse, rat, and human healthy volunteer populations available. And then you have your compound data or substrate. I'll go into the details of these screens later. And the third element of the simulation is the trial design. So I'll go through each of these in a little bit more detail. So on the kind of the demographic or the species screen, you have your CYP phenotypes, transporter phenotypes. The names have been genericized here because the screens are the same across species, but with the annotations you have the details here for 2C9, 2D6, 3A4, and there's further, if you want to change this information, there's more information available in the help files to change that. On the liver and the GI tract, obviously it has the relevant physiological parameters, including enzyme abundances, intrahepatic recirculation, and gastric emptying time, etc. Key to note is all the data that's available in the species side or the systems parameters is already been collected and through rigorous scientific research. So you don't have to worry about collecting or inputting any of this data unless you specifically want to change a particular parameter, for example, gastric emptying changing time. And when you And the other key element is when you switch through species, only the kind of the values of the parameters changes. The screen looks exactly the same. So it helps when you're switching between preclinical and human species in terms of development at the early stages of discovery. So that was one of the key elements of our of improving the user experience, having animal and human species together. And then you have other parameters like the tissue volume and blood flow compositions, flow rate, and the brain compartmental, the brain paraphysiological parameters. Moving on to the substrate side or the compound side, which is obviously very important from a sponsor's perspective. So by default, you have a midazolam file available for each of these species. So that model is there. And this is the FISChem and the blood binding parameters screen. So here you input your FISChem parameters, and wherever you see this kind of calculator icon, if you click on it, it shows you the calculator for that relevant parameters, and obviously you can switch between other attributes as well. Then in the absorption side, as Mark mentioned, you have two options: the first order absorption model and the advanced ADAM model. So in the first order, so in terms of your FA calculations, you can either enter it yourself, or you can predict it using the mechanistic permeability model, or MECPEF model as we call it, or the PAP using CACO2 or MBCK cells, etc. So just to show you the ADAM model options, ADAM model is where you can look at different formulations. So you have various different oral formulations options available, like solution with precipitation, suspension, solid formulations, like immediate release, entry coated, or you can define your own disintegration profile. Just to kind of show you, so you can have a discrete function there or input data, as well as you can also specify your own dissolution profile if that is something you have the data for, so both for stomach as well as your GIT. And then you have other options like control or modified release capsules or granules. Here for control or modified release, you can even have a formulation gastric emptying time if it is different from your physiological gastric emptying time. So you can change that here. And then you have other options like the diffusion layer model, where you can account for intrinsic solubility or at a specific given pH, or a discrete solubility pH profile. So you can enter that here as well if you have a differential solubility across a range of pH profiles. The other aspect is you can also account for biomediated solubilization enhancement and functions such as that. So it has a lot of different options for simulating oral formulations. Moving to the distribution tab, as Mark said, you have three main options: the minimal PBPK model, the full PBPK model, and the compartmental model, which is the 1, 2, or 3 compartmental model. You can also fit the 3 compartments here on the screen itself, if that's something you want to explore. In the full PBPK model, you can predict your VSS using a couple of different methods. The default is the Rogers and Rowland's method. The details obviously are there in the notes here and in the help files, of course. And using that, it predicts the tissue plasma partition coefficients for each individual tissue. In the elimination tab, you have your in vivo clearance, so CLPO or CLIV, options for whole organ metabolic clearance, either measure Clint values measured through liver macrosomes or hepatocytes. So you can either enter Clint or a Vmax KM value. In the enzyme kinetics tab, again, you can specify that based on the specific enzymes that you have measured in vitro. And the calculator icon here is essentially to kind of predict your additional clearance fraction if that's of interest. In addition to your enzyme kinetics, you can also account for additional clearance which has not been accounted for the enzymes mentioned here, in plus biliary clearance as well as a renal clearance component. So there's the options available. Then when you have the full PBPK model selected, you also have the option to simulate transporter impact. So you have options for intestinal, apical, and efflux and uptake transporters. Within the liver, you have your sinusoidal efflux and uptake transporters and canalicular efflux transporter. And within the brain model, you have a 5-compartment brain model for which you have multiple transporters available across each brain interface. So you can account for that as well. Then you have your PD options, so you can look at both PK-PD relationship here, and you have options for a simple Emax or a direct response model or an indirect response model, as well as functionality to write your own custom PD code if that's of interest. So that's on the substrate side. So moving to the trial design. So as Mark said, you can look at both the species representative as well as a virtual population. In terms of the trial duration, it can go for multiple days for a single or multiple dose, as well as on fasted and fed condition. Now, I will just quickly load a workspace. So a workspace essentially is a file that kind of saves your species data, substrate data, as well as your trial design parameters together, and you can use this for either sharing between your colleagues or sharing where for later. So in this particular case, this is a trial simulation for 56 individuals over 6 days for a sample drug, and it's a multiple dose given every 8 hours up to 3 doses. So that's the trial. In terms of the outputs, the default output is Excel. So you can then select into each individual kind of sheet that you want to output. So I've selected some relevant ones. I'll show the examples there. One of the interesting options here is you can overlay an observed data file. So I've selected a sample observed data overlay, which I'll show you the in just a second. And when you run the simulation, it will output the Excel. So for the interest of time, I've generated the Excel here. So this is your summary sheet, which kind of captures all the technical information as well as the input parameters used for the simulation. So it's essentially for audit keeping purposes. You cannot change any of the attributes on the screen here. Same goes for your input sheet and your population data. So if you're using this for regulatory submissions, this is what they would refer to to understand what you have done. So the key element is the concentration time profile. So by default, you are given the mean concentration time profile and the 5th and the 95th percentile. So this is what I've shown on the graph here. And then the dots here are the observed overlay files. So as I'd shown before, so this is quite neat. We are able to capture almost the observed data file. And in terms of these, the individual population side, so you have the individual concentration time profile for all 56 individuals here. So you can, obviously dig deeper into any of the particular individual if you wish to. So in addition to that, I want to show you the regional, because we have used the ADAM model. So this is the regional fraction of dose absorbed in each segment of the gut, plus the fraction metabolized in each segment as well. So that gives you an idea on that. And when you have the full PBPK model, you can look at the individual tissue concentration time profile as well. So for example, here you have the adipose tissue, your brain concentration. So this is useful when you are looking at a tissue-targeted drug profile and whether the drug is reaching therapeutic concentration as a particular dose within that target tissue or not. For example, brain, kidney, or muscles, or some other tissue. So that's what I wanted to share. So I'll hand it over back to Mark.
Thank you, Himanshu. We have a few questions to go through to potentially have answered today. So just 1 second, I'll pull those up. So first question. Oh yeah, let me. I guess I'll go ahead and show my camera too. There we go. All right. Okay, we're all on screen now. So the first question. If I want to evaluate Simcyp Discovery, do I need to install it locally on my machine? Kevin, you want to take that one?
Sure. We have a hosted cloud-based installation that we're able to provide you so that you can do an evaluation license for approximately 2 weeks. That way you don't have to necessarily work with your MIT department or others to get it going, so we can get you going early, and we certainly will support you throughout that process.
Great, thank you. Next question. What kind of training and support do you offer? Himanshu, would you like to take that one?
Sure. So we have dedicated workshops for different kind of Simcyp functionality. For discovery, we have what we call as the first in human workshop, which uses the discovery simulator and its specific examples. And we run that every so often throughout the year. So you have the option to attend that in person or virtually.
Great, thank you. So the other question is, why should I buy Simcyp Discovery relative to some of the other PBBK packages? I'll go ahead and take that one if I may. So it's fully compatible with the leading PBBK Simcyp simulator with regard to importing of model files. Also, it supports seamless translation across animal species to humans, and its ease of use is it's ease of use and is cost competitiveness for the needs of preclinical teams. And then finally, you can do some initial drug-drug interaction screening using the static DDI calculator. And then we also support deployment of the platform either via, on desktop installations or on servers for remote access. So next question. Kevin, maybe you can take this one. How do you get started using the tool if I have no experience with PBBK modeling?
Yeah, we have a lot of different arrangements that we can do with you. First of all, we could, because we have a consulting organization also, we could arrange projects together that would allow you to work on a project with our team. As we mentioned, we do have these workshops that allow you to get a level of education, and then you want to springboard from there to go into a project as quickly as possible. And the best opportunities are understanding what your needs are and helping us to understand what your level of capabilities are and what additional support you would need from us. So we have a variety of ways that we can approach these, and we just look forward to meaningful discussions with you one-on-one to really understand your needs.
Okay, our last question for the day is, and maybe Himanshu, you can take this one. What types of clients are using Simcyp Discovery?
Yeah, so currently we have kind of various clients in the biopharma space using Simcyp Discovery, and a few CRO folks as well looking at the, evaluating Simcyp discovery as we speak. And it's mostly around the early discovery and development programs, either to predict the FIH dose or for formulation assessment. Kind of internal decision-making point as to which formulations to go for. Yeah.
Okay. Well, I want to thank everyone for attending today, and I want to thank my co-hosts Kevin and Himanshu. As we have no further questions, I think that will be all for today. Trying to remember, is Sebastian, did you have any closing remarks?
Did you want me to take? I'll take that, Mark, for the closing remarks.
Yes, thanks, Kevin.
All right. So thank you again for joining us today. As you heard from Mark and Himanshu, over 100 novel drugs and over 300 label claims have benefited from our Simcyp modeling technology for regulatory approvals in lieu of clinical trials. And the Simcyp discovery is derived from this top mechanistic predictive modeling platform and allows you really to use it across the whole drug development process. We've got a series of use cases that are really the focus of the initial application: first in human prediction, DDI screening, looking at trade-offs between compounds and formulations for your candidate selection, and to be able to look at cross-species predictive analysis. As we mentioned, there's a lot of options out there, but if you really want a smooth transition from the discovery phase into the clinical phase, being able to use one technology from Simcyp discovery to full Simcyp really will support your growing needs without having to input and export data sets. We've tried to make this, we've made this as easy as possible for your utilization for your needs. It could be as simple as somebody working off of a laptop, or as your needs grow in the organization, number of users increase, that we enable you to work off of a server construction so that you can run remotely, you can run your simulations remotely. As we talked about before, when you're looking at the opportunities for getting started with this, we do have a team of scientists that are using these tools to consult on companies or initial projects. And so these tools have been used under the rigor of very strong PBPK modelers, and they're available to work with you or to help consult to you as you get your capabilities up in-house. So we look forward to meeting with you and understanding your specific applications, and wish you well with your future drug developments. Thank you for joining us, and have a wonderful day.
Oh, Kevin, a question has just come in, actually.
Okay.
Yep. The question is as follows: do you have a multi-user license for Simcyp discovery?
So our license is our initial desktop platform allows it to be used by multiple users in physical access to the platform. The server one does allow you to remotely run your simulations from a server that would be hosted within your environment. So that, yes, it does allow for multiple users. And what we'd like to do is understand your user needs and set up the right configuration for you.
Okay, thank you, Kevin. I think that is all the questions we have for today.